and my chemo worked. They went to the clinic. Phase 1, phase 2, you will see how expensive all this happened. Some billions, maybe 5 billion has been spent, and the asthma patients did not respond. Can you imagine? A very small fraction of the patients responded. The drug is not in the market. They are still trying to find out why it's not working. Because the hypothesis was wrong, because the animal model is dependent on eosinophilia. Removing eosinophilia helps a little bit, but still patient cannot breathe. You can clean the lung, 99% cannot breathe. The basic problem stays. So I'm now bringing the problems, okay? So you choose a better one today, IL-13, okay? That's what is happening. People have anti-IL-13 antibodies, it's in the clinic. It will not work. I know. I'll tell you why I know. But anyway, you select such, among all these unknowns, a target, and then you say, I have an assay, molecular assay or cellular assay, then you have a lot of compounds, either computationally or classical medicinal chemistry way, you synthesize thousands and thousands, up to a million. Big companies have more than a million compound collection. And nowadays you don't do, researchers don't do screening. It's another department, it's robotics, you just arrange everything. A million is screened within 3 or 4 months. Then you get hits. What do you do with the hits? Then you prove that this is really hitting your target. Then some of the hits are good looking, you can change them, you can make them better, more active potency increasing. This is called lead optimization. You do all that, now the team composition changes. And slowly you select a candidate. You say, chemist, now you should start working, changing the right hand, left hand, that many. It becomes more toxic, less toxic, more bioavailable. There are so many parameters. So you have to synthesize up to 1,000 to 10,000 compounds from a single hit. Among that, one of them will become a drug. Okay? And that goes to preclinical, pharmacological, toxicology, absorption, distribution, metabolism, excretion, all that has to be done in the animal. And also genomics, whether it's hitting any DNA or not. All this is done, and you lost already, we'll see how many years? 12 years minimum. And then you go to the clinical studies. And there are 4 phases, okay? And by the time you go and say, look, phase 1 work, 2, 3 work, I want this drug to be registered by an authority like FDA, for Food and Drug Administration, and Europe is EMA, I don't know what they call it. But, and then if they approve, of course they are competing. Many different companies have similar compounds. Yours has to be superior to the other one, right? Not toxic, good, then it's approved, then you can start selling. A patent has to be done right here. If you patent very early, your clock starts working. You have only 15 years to protect your drug. Okay? And therefore, early patenting is not good. But if you are late, somebody else discovers exactly the same, you lose the whole game. So there's some time frame you decide and patent, protect, so that nobody can sell the same drug, because you spend all this money here. We'll come to the money business later. Then, let's, before going to the market, look at this here. This is clinical study. Phase 1. I don't know if you've got any MD except you. There are MDs here. You know all this, right? You have been participating maybe. Phase 1 is the first time in man, it's called. And what you do, you take 20 medical students usually, because they're available, they understand, they get money, they're poor usually. So you give them normal healthy people a drug, not test whether the drug is working or not, but rather increasing doses, checking the safety. and determining what side effects the guys or the ladies are getting, right? This is $1 million or $2 million sometimes cost. Phase 2, 100 to 300 people is given same drug. First time you are asking the question whether or not my drug is hitting the target. Proof of concept. Very important. In man, you are asking the question, I want to inhibit enzyme X. Am I inhibiting that or not? If yes, that's good news. If not, drugs is shaky situation and it may go to other directions, it may die altogether. Okay? If it is hitting, then you go to the next one. This is really expensive and also a very crucial stage because you are testing now, depending on the drug and the field, up to 3,000 people. Each one gets money, doctors get money, hospitals get money. This is very expensive. A small company cannot do this. Usually, small companies making discovery is bought by bigger ones because they have the money. Okay? It goes into the clinic and then you confirm that it does what you think it should be doing, but also you discover side effects, manageable or not, and collect a lot of information about the drug because it's a huge number of patients. Okay? The phase 4 is you are now in the market, everything is accepted, but still it's going from 3,000 to 10,000 to 100,000 to 1 million is a very different issue because of the genetic heterogeneity. There are drugs, I had witnessed one of them, and it caused the company to merge with another one in order to survive. An allergic drug hit the heart. 4 patients died, it's phase 1. The company has to merge because they were hoping, Salzane, name of the drug, Aventis. It became Aventis. It wasn't. It was HMR earlier. You can change the history of a whole successful company just because it prolonged QT time. And these you understand what it done. And the patients, these 4 patients, were getting other drugs. Okay? So there is drug-drug interaction, which I didn't even mention. And that happened up to 1 million times. You know, when you go to the million, you may have a side effect that you will never see in 100,000 patients. So I'm trying to give you a feeling why drug discovery is difficult. Here is another problem. There is a paradox in drug discovery because companies pour money like mad. This became 200 billion in the United States now. This is an old slide from FDA. But as you put more money, you should be getting more and more drugs, but you don't. Okay? Actually, it looks like it decreases. At this 2007, there were 20 new drugs accepted by FDA. But people put $120 billion, they increased. So you're putting more money, you're not getting more drugs. It actually is flat since 2011. It is flat, but that number is increased a little bit. You're getting 25 to 30 drugs per year now, okay? But you're putting up to $300 billion only in the United States. These numbers are really, really big. So there's something wrong in this whole process. We are wasting time, or we don't know something. We are doing something wrong. And this something wrong will come here even more pronounced when I tell you a big company, by the time a drug goes into the market, spends $500 million or $1 billion. And this is 15 years. When you put it together, the shortest is 12 years. You see how crazy investment is this? It's like actually putting your whole life raising a kid or having a kid. How dangerous in this world to have a kid. And this here is summarized, I think. I give you a little glimpse why it's taking too long, why it's so expensive, and why we are not getting, no, this will come, why we are not getting more drugs by putting more money into it. In Trian, when I moved there 2003, there were some clever people except me. These guys were wondering why we are unsuccessful. This company, 100,000 people work for the company, and they are known for tapes and a lot of very intelligent products in aeroplane industry. Okay? Excellent company, but they had a secret pharma division, just 2,000 people. And I became one of them. And we started wondering, can we do better? We should be more clever. Don't make the mistakes that other companies are making. Trian is very good academy for that. We spent 8 weeks, hours, put together. These guys are very intelligent guys. Some of them are chemists, some of them are biochemists. There's only one immunologist here, exception. Anyway, we discovered the reasons for the mistakes is that the managers have to make decisions in the face of many, many unknowns, like a politician. You say something, I will do this and that, but you have a lot of unknowns. You have to have a good instinct, gut feeling to do the right guessing, okay? So that's the managerial difficulty. Then we discovered that mistakes that are made at the beginning, for example, conceptual failures. You put all your money thinking that the asthma or allergy is happening because if you are wrong, all these 15 years and drug will not work in the end. Take rheumatoid arthritis. There are 30 years I work on T-cell inhibition for rheumatoid arthritis. Because animal models, when you remove the T-cells, a particular kind, beautiful therapy. In man, finally, after 30 years, people discover it's not a T-cell. By the time a patient sees a rheumatoid arthritis, it's not a T-cell disease. Without the T-cell, you don't get the disease, but the patient already had the complication. Pathology is full, and it's T-cell independent. We didn't know that. It's B-cell dependent. That's what we think now. But I doubt, in the end, anti-TNF, 70% of the patients respond. By chance, not by thinking. By chance, TNF is successful. That's the whole answer. There's a huge research going on here on rheumatoid arthritis. TNF is a good thing, anti-TNF, but there are, I'm sure, better, because still we don't know why we get rheumatoid arthritis. Again, basic science. I'm so glad to hear from Şefik is that the Koç University is putting a lot of effort for basic science in every field. That's the most crucial one. Another thing we discovered, I like these numbers. Sometimes when you play with the slides, the numbers change. Nobody needs an expert. Everybody knows things better than anybody else, especially if you're a manager. You're supposed to know, right? I was a manager. And we don't call independent researchers, come and look at this. Because you have people treat their ideas like they're babies. People treat their teams like their family. This is a human nature. That means they're protective. They tend not to see the failures. They tend not to accept their concept is wrong from the beginning. Okay? They become defensive. You need independent people coming and looking at all the projects and saying, this project cannot work because they have their scientific reason. This project has a better chance. That's what we do. But before doing that, I'll come to that detail. Before doing that, we discuss